Hello, everyone. My name is Juliet Morrison, and I'm an assistant professor at University of California-Riverside, and I'm in the Department of Microbiology and Plant Pathology. And today I will be presenting exciting results from my group that show that pleural macrophages promote recovery from influenza. So let's get started. Influenza has been with us a very long time. It was described as early as 400 BC. And major epidemics and pandemics of influenza have occurred throughout history. And many experts believe that another influenza pandemic that could kill millions of humans is inevitable. There is also a seasonal burden of seasonal influenza. So seasonal influenza results in 3 to 5 million cases of severe disease and 290,000 to 650,000 deaths annually. There's also a very high economic cost, with 11.2 billion in economic costs per year in the US related to influenza. Like other respiratory viruses, influenza virus is transmitted via droplet transmission. The virus enters the respiratory tract and attaches to ciliated epithelial cells lining the sinuses and airways. Then, as the virus replicates, the cilia are destroyed. Mucus cannot be removed, and so the airways become clogged and stimulate coughing. And destruction of cilia contribute to secondary bacterial pneumonia infections, sinusitis, and otitis. Now, what we study in the Morrison lab is the host response. And what is the host response? It is the reaction of a living system to a stimulus. And the disease severity of a viral illness is determined by the interplay between the virus and the host response. If you have too little of a response, that's bad, because then the virus will replicate unrestricted, and that will cause a lot of tissue damage, and so you get disease. But you can have too much of a response, and that is also bad, because if the response is too exuberant, you end up with a whole bunch of other tissue damage of healthy tissue, and this can also lead to disease. So one of the goals of the Morrison lab is to develop host-targeted viral therapeutics. And so what we envision and imagine is that there is a balance between viruses and the host response. And so we look at it as a seesaw. And so what I'm showing you here is an image of children on a seesaw. And they are representing viruses versus the host response. And in this image, we have the viruses, and they're winning. So you can see one red arrow here where the viruses are clearly winning. But if you were to look at the other red arrow pointing to that child in the red hat, if we were to just move him over slightly, we could balance that seesaw. And that's what we're aiming to do. We don't want to just dampen the whole immune response. We would like to find specific cell types and pathways that we could target that could allow us to tweak the immune response so that we can still fight the viruses, but prevent the negative consequences of an overly exuberant immune response. And so for one of our cases that we're interested in is influenza. And one of the things that we were trying to do is to figure out how we can take a host-targeted strategy for treating influenza infections. And to do this, we went ahead and compared the host response to different influenza viruses. And so we asked the question, is there a long gene expression signature that can distinguish mild and severe influenza? So to do so, we use two data sets. One here from an older data set that I published in 2014, and another similarly designed one from 2012 from Jossett et al. And what we have here are biopsy mice that have been infected with different strains of influenza. a wide range, in fact. So we have H7N9, avian influenza, H7N7. We have H5N1. We have the pandemic H1N1 from 2009. And then in the Josset et al., we have a range of H1N1 seasonal influenzas, the 2009 H1N1 pandemic strain, and we also have the notorious 1918 reconstructed influenza. So we have a wide range of viruses with a wide range of outcomes. So I'm pointing you to this graph here, where on the y-axis we have percent body weight, and on the x-axis we have days post-infection. And you can see that the outcomes fall into roughly 3 groups. So we have here our seasonal flu viruses that cause a mild disease. The animals lose a little bit of weight, but then they recover. Then we also have some animals that lose a lot of weight, but then do recover. And then we have other animals that lose so much weight that they have to be euthanized, or they die naturally. And these animals fall into our group that are infected with our avian influenza viruses or the 1918 influenza. So we have this wide range of viruses. And then in both of these studies, we also have viral titers, we have histopathology data, and we have gene expression data from whole lungs. And the aim for this study was to take all of this data, put them together, and to identify tissue signatures and predictive markers. And that's what we did. So we found that pathogenic viruses induce a specific gene signature. So let me walk you through these data. So here I have this large heat map, and I want to point you to the top of the heat map here where I'm showing you percent weight loss. So I have all of these viruses here lined up from left to right, from least pathogenic to most pathogenic, based on percent weight loss. So you can see that lined up there. And then you can see that on the left, we have different modules, different gene modules. And you can see here, let me move forward. That in one module, the inflammatory red module, we can see that for the inflammatory responses, that we have more expression of inflammatory genes with viruses that are more pathogenic. So viruses on the right. We also found that for genes that are involved in metabolic responses and tissue repair mechanisms, they're less expressed in the lungs of animals that were infected with the highly pathogenic viruses and the 1918 strains. What was very interesting to us, though, were these modules in the middle, these lipid metabolism, coagulation signaling, oxidative stress responses, etc., that were more highly expressed in the lower pathogenic viruses versus the higher pathogenic viruses. And so we asked the question, what was actually going on there? What cell types are actually driving these differences? And we know that these are whole lungs. So the lung is made up of a mixture of different cell types. We have epithelial cells, we have different immune cells. Which lung cells are actually responsible for these transcriptomic differences? To get a feel for which cells are actually responsible for this, we decided to take a systems level view. And so we employed this tissue deconvolution algorithm called the digital cell quantifier, or DCQ. And what DCQ does is to take genome-wide gene expression data, like what we have for our whole lungs, and combines that with an immune cell compendium from ImGen, which has a bunch of different immune cell types. and then takes this, puts it together so that it can infer an in vivo global dynamic change in the immune cell quantities within this complex tissue, like the lung. And so in using DCQ along with regression analysis, we were able to identify lung populations that correlate with weight loss. For example, so once again, I want you to take a look at the top of this figure where we have our weight loss. So we still have our strains here lined up from left to right. We have our least pathogenic to our most pathogenic outcomes. And we see that there are various immune cell types that are correlated with disease severity. For example, we have granulocytes that are predicted to be enriched in animals that have more severe disease outcomes, so more weight loss. And this is actually known in the literature that when you have more neutrophils in the lung, you have more severe disease outcomes. DCQ also predicted that there would be less alveolar macrophages in animals that had more severe outcomes, which is also known in the literature. But we weren't interested in using DCQ as a validation tool. We were interested in using it as a tool for discovery. So we were very interested in a particular population that had popped up and that we had never seen before. And that population was this small peritoneal macrophage population that we saw here. And it was very curious to us because we asked, what are peritoneal macrophages even doing in the lung? More importantly, what we noticed is that this population was predicted to be only in animals that survived infection. So this is very exciting to us. Here's a population of macrophages that are supposed to be in the lung, but only in animals that survive influenza infection. What's going on here? So this is where the hypothesis generation begins. So we asked ourselves, peritoneal macrophages, where are these found? Well, these are found in the peritoneal cavity. And so what is the peritoneal cavity? Well, the peritoneal cavity is a serosal cavity, and the serosal cavities are the peritoneal cavity, which surrounds our abdominal organs, the pleural cavity, which surrounds our lungs, and the pericardial cavity that surrounds our heart. And so these are fluid-filled cavities that protect and cushion our organs, but also contain immune cells, primarily macrophages. And so because of the relationship between the pleural cavity and the peritoneal cavity, we hypothesized then that what we were actually seeing in the lungs was actually pleural macrophages and not necessarily peritoneal macrophages. So serosal cavities contain similar cell populations. They contain macrophages, and serosal macrophages are divided into two populations: small macrophages, which are MHC class II positive, F480 negative. They are smaller than the large macrophages, and they also are about 10% of the macrophage population. We also have our large serosal macrophages, and they are MHC class II negative, F480 positive. They make up about 90% of the population, and they are larger in size as well. And so we thought perhaps then macrophages that we were seeing that were being predicted to be in the lung in our surviving animals were our small serosal macrophages, and specifically small pleural macrophages. So here's our hypothesis. We hypothesized that pleural macrophages were leaving the pleural cavity, crossing the mesothelial layer, and entering the lung to do something, perhaps inhibiting influenza replication or inhibiting damage but doing something to help promote recovery from influenza. So we went ahead and decided to test this hypothesis, and this project was driven primarily by a graduate student in the lab, James Stump. And he started off by testing whether or not we could label pleural macrophages in vivo. And it's notoriously difficult to label, to do intrapleural injections, but we've actually worked out a very good technique. And so James was able to inject the pleural cavity with PKH dye, which specifically labels phagocytic cells, and then he harvested these from the mice that have been intrapleurally injected. And you can see here in red that the macrophages have picked up the dye. So yes, we are able to label these macrophages, and we can do it in vivo. I also want to show you here what happens when we set up an influenza virus infection using our model. So here is an intranasal infection with California-09, which is a strain of influenza that we use. And you can see here on our y-axis, we have our percent of initial body weight, and on the x-axis, we have days post-infection. And in this particular, with the particular dose that we use, we have a little weight loss, and then we have recovery. So this is a model of a mild disease from which the animals recover. So then we ask the question, do pleural macrophages traffic to the lungs during influenza? And so on day one, we intrapleurally inject the PKH dye to label the macrophages. And then on day two, we do an intranasal infection with Cal-09, or we do a PBS control. Then the lungs are harvested on days 6, 9, and 12 post-infection. And then they are used for flow cytometry. I just want to outline how we sort our pleural macrophages as well as our lung macrophages. So for our pleural macrophage sorting, we're taking our single cells that are alive, and then we are looking at CD115 and CD11b positive cells, and this is all of our pleural macrophages. And then we are distinguishing them into small pleural macrophages if they're MHC class II positive, and large pleural macrophages if they're F480 positive. Now for lung macrophages, we're taking our singlets that are alive, and we consider them macrophages if they're MERTK positive and CD64 positive. And then we are then differentiating them into two populations. Interstitial macrophages, we consider them interstitial macrophages if they're CD11b positive, and we consider them alveolar macrophages if they're Siglec-F positive. So now for the questions. Do pleural macrophages traffic to the lungs during influenza? And so here are results for day 6, day 9, and day 12. I want you to take note of the axes. We have our live cell numbers from our lungs, and we are looking here at what happens in our PBS mice versus our Cal-09 infected mice. And both of these mice have been labeled with PKH intrapleurally. And you can see here that only in our California-09 mice do we see the presence of these PKH positive cells accumulating, and they peak on day 9 post-infection. I also want to reorient you to this map that we have here of the weight loss. pointing out specifically when weight loss occurs, which is on day 6, and when we start seeing weight gain, which is on day 9. And so when we're seeing the accumulation of these pleural macrophages within the lung is when we're actually seeing the animals starting to regain weight, which would suggest that these pleural macrophages are involved more in the resolution of disease, in the resolution of disease. We also asked what these PKH positive lung cells were. Are they alveolar macrophages? Are they interstitial macrophages? And we found that these PKH positive cells are actually CD11b positive, indicating that they are interstitial macrophages. They're not alveolar macrophages. They don't have cyclic F. We also asked, where are these PKH positive lung cells? So here we have some microscopy images of the lungs. In red, we have our PKH stained macrophages, and in blue, we have DAPI, which is staining all the cells. The top panel shows our PBS animals, and so we don't see any red because there are no macrophages going into the lung. And on the bottom panel, we have our Calo9 infected animals, and you see the accumulation of these pleural macrophages in the lung near to the mesothelial layer. So the little dotted white line there is the mesothelial layer. So pleural macrophages accumulate in the lung near the mesothelium. So we then asked, do pleural macrophages impact influenza outcomes? So it's great that they're going into the lung, but are they even doing anything? Are they even important? So to test this, we did an intrapleural injection of clogenate liposomes. So what this is that clogenate basically is a poison, and the liposome will direct this poison specifically to phagocytic cells in the pleural cavity. And so when you inject these clogenate liposomes, the pleural macrophages in the pleural cavity will pick this up, and then they will undergo apoptosis and they'll be depleted. We also have PBS liposomes as a control. And the first thing we did was to look at the levels of virus in our lungs post-infection. And we looked at day 3 and day 6, and we found no difference in the viral loads in mice that had pleural macrophages versus mice that lacked pleural macrophages. So there was no impact on viral replication. And so on our y-axis, we have plaque forming units per gram. On our x-axis here, we have our PBS liposomes versus our clogenate liposomes. And we also looked at at weight loss. And so on our y-axis here, we have percent of initial body weight. X-axis, we have days post-infection. And here we actually see a difference. You can see that we have increased weight loss in the animals that had the intrapleural clogenate liposome injection, which indicates that when you deplete the pleural macrophages, there is increased weight loss during influenza virus infection, showing that the pleural macrophages are important for influenza virus outcomes. And to get to the root of what's going on here, we went ahead and looked at what was going on with the immune cells in the lung. So when we looked at the total lung CD45 cells, so all the leukocytes in the cells in the lungs, we didn't see much of a difference in the PBS-L versus the clogenate liposome treated animals. But when we dug down and looked more deeply, we found that when we looked closely, there was an increase in the amount of neutrophils in our animals that had that lacked pleural macrophages. So having a lack of pleural macrophages led to an increase in the amount of neutrophils in the lung. And as I had mentioned earlier, one of the things that we do see in the literature mentioned often is that when there is a high amount of neutrophils in the lung, that is often associated with severe influenza. We also found that by doing ELISAs, we found that the pleural cytokines were different as well. We had increases in interferon gamma, TNF alpha, and MCP1 in our animals that lacked pleural macrophages. So overall, pleural macrophage depletion causes increased neutrophil influx into the lung during influenza virus infection. So to summarize, I've shown you today that we used whole lung transcriptomic data and bioinformatic tools to identify immune drivers of influenza outcomes. So we were able to actually take transcriptional data and use it to generate hypotheses. And our data indicate that pleural macrophages migrate to the lung during influenza virus infection. So pleural macrophage depletion does not affect lung viral loads, so it's not affecting viral replication, but it does impact disease outcomes. So it does increase, so pleural macrophages are important for disease outcomes because depleting them increases the disease severity by leading to an influx of neutrophils into the lung. And finally, pleural macrophages improve influenza outcomes. And so I'd encourage you, if you are interested in learning more about this project, to check out our preprint on bioRxiv that we posted last month, and we're currently working to submit this formally. And I'd like to acknowledge the people that made this happen. This project was primarily driven by James Stump in my lab. And I'd also like to thank the members of my lab, the Nair lab, and at UC Riverside, as well as others at UW Medicine and Tel Aviv University. And I look forward to hearing your questions and comments. Thank you so much for listening. Have a good day.